Dear students, please note down. The PDF file of all these notes have been uploaded in the description of video lecture. Please download your notes. In today's lecture, we will discuss sliding filament theory of muscle contraction. Dear students, it is a common topic for all competitive exams and BSc students. BSc students, please note down. This is your final exam topic. Sliding filament theory of muscle contraction, and this theory was proposed by A.F. Hagsley, H.E. Hagsley and Hansen in 1954. As we know, in muscle contraction, sarcomere is the structural and functional unit of muscle contraction. Dear students, sarcomere. And that sarcomere, this is the ultrastructure of the sarcomere, which is structural and functional unit of muscle fibers. And in this sarcomere, a complete band, A band, which is anisotropic, and half I band on both sides of this A band. Dear students, in this sarcomere, this A band which is complete, and on its both sides of this A band, half I band and half I bands are present. This A band, which is composed of both thin and thick myofilaments. Dear students, A band having both thin and thick myofilaments, and in the center of this A band, a zone is present which is H zone, H line, Hansen zone, Hansen disc, and in its middle part of this H zone, a line is there which is M line, which is Mittler's chip, which is middle line in German language, Mittler's chip. The free ends of thick myofilaments, these free ends of thick myofilaments which possesses heads or cross braces. Dear students, A band, and in this A band both thin and thick myofilaments, free ends of these thick myofilaments which produce cross braces or heads, and in this A band, H zone is present, Hansen line is present, and its middle part M line is there, and these I bands, isotropic bands, these are made up of only thin myofilaments. Dear students, thin myofilaments, as we have already discussed, thick myofilaments are made up of myosin protein. So these are myosin filaments, and these thin myofilaments are made up of actin protein. So these are actin filaments. I band, only thin or actin filaments, while in these A bands, both actin and myosin filaments are present, and the free ends of these thick or myosin filaments which produce cross braces heads. This one is H zone, and this middle line, that is M line, and the interval between two successive Z membranes, and these Z membranes are present in the middle of I band. This interval between two successive Z membranes, this is sarcomere, which is structural and functional unit of muscle fibers. Dear students, today's topic is sliding filament theory of muscle contraction, and that was given by Hagsley and Hansen in 1954. Its features are, first point is during muscle contraction, thin myofilaments slide inward. Dear students, note down, these thin myofilaments slide inwards towards this M line, H zone. First point is very clear. During muscle contraction, these thin myofilaments slide inward towards H line. Second point is during muscle contraction, the sarcomere, it means the interval between two successive Z membranes, this sarcomere, its length shortens. It means both Z membranes come close to each other. Point number third, the cross bridges of, as we know, the free ends of these thick myofilaments which produce heads, which produce cross bridges. These cross bridges interact with these thin myofilaments, and these cross bridges pull the actin myofilaments towards H line. Point number next, as the thin myofilaments move past the thick myofilaments, the H zone may disappear. Dear students, as these thin myofilaments which move towards this M line, as a result, this H zone which may disappear completely. Next point is the length of thick and thin myofilaments. Point. Dear students, note this point which is very important. It means the length of this thin and the length of this thick myofilament during a muscle contraction. Length of both thin and thick myofilaments, that length remains constant. It means during muscle contraction, very clear, the cross bridges, the heads of these thick myofilaments which interact, which bind these thin myofilaments, and as a result these thin myofilaments which move inward towards H zone. As a result, this H zone may disappear completely. Dear students, this H zone, H line may disappear completely. Both Z membranes, these Z membranes come close to each other. As a result, the length of this sarcomere, very clear in this contracted state of muscle, this sarcomere length that gets shortened. H zone completely disappear. And dear students, note down, length of this thin as well as length of this thick myofilaments, this length remains constant. In this mechanism, sliding filament theory, heads, cross bridges of these thick myofilaments, and these thick myofilaments are made up of myosin protein which interact with the actin proteins of thin myofilaments. So they produce a complex, and that complex is actomyosin complex. And the heads undergo, these cross bridges or heads, they undergo rotation. As a result, these cross bridges pull these thin myofilaments towards H zone. As a result, this H zone may disappear completely. And dear students, in resting state, the sarcolemma, the plasma membrane of a muscle fiber that is polarized, as on its outside positive charge is there and negative charge inside the membrane. inside the sarcolemma, inside the sarcoplasm. And this potential difference, it is resting potential. At this time, on this sarcolemma, a pump operates, which is sodium potassium active pump. And this sodium potassium active pump, which pumps generally 3 sodium ions outside and 2 potassium ions inside. It is an active pump which maintains a polarity, a potential difference over this membrane. It is resting potential, and the membrane at this stage, it is polarized. And this polarized membrane, in response of a stimulus at the time of muscle contraction, this polarity is changed. It becomes reversed. So, negative charge develops outside and positive charge inside. At that time means at the time of muscle contraction, this polarized membrane becomes depolarized. Dear students, this is all about the mechanism of muscle contraction. It is sliding filament theory. In this entire process, the cross bridges the heads of thick myofilaments, myosin filaments, which interact with these thin actin myofilaments. These cross bridges undergo rotation. As a result, these cross bridges pull the actin myofilaments towards H line. As a result, this H zone may disappear completely, and the length of this sarcomere that gets shortens. But the length of this thin and thick myofilaments, both thick as well thick myofilaments, that length remains constant. This is all about sarcomere, sliding filament theory of muscle contraction that was given by Hexley and Hansen in 1954. It is very important topic for all competitive exams and dear students, BSc final exam topic. In tomorrow's lecture, we will discuss another important topic, that is Cori and Cori cycle. Thank you.